Hello everyone, today we'll learn about the structures and the common derivatives of the monosaccharides. So if you are new to biochemistry or if you are from a non-biofield or you are a beginner in this field, then you might find it very confusing or it might find it very difficult to learn the different structures, the linear form, then the closed ring structure, the different derivatives, the different forms, the different epimers. So the structure, there are minor differences and it is very difficult to remember all the structure. In today's lecture, we'll try to simplify and learn all these structures so that it becomes easier to understand the biochemistry. It becomes easier to understand the disaccharide and the polysaccharides like structures, and even at undergraduate level it is often asked to draw the structures of these sugars, so you can easily draw. We'll try to learn all these things in today's lecture. Now let's begin with this. Now okay. So in first we'll learn about monosaccharide structure. In monosaccharides, the simplest of the monosaccharide is glyceraldehyde. 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 So we have learned in the previous lecture that it is a triose. Triose means 3 carbon and it is an aldotriose. Aldotriose means it is having an aldehyde group. So let's draw the structure. So it is a triose. I have drawn 3 carbons. First, second, and third. The labeling is always done from this end, from the heaviest functional group. So because it is an aldotriose, so it means it has an aldehyde group. So it is CHO, and then this is H, H, and this is CH2OH. And if we talk about its respective keto sugar, that is dihydroxyacetone, DHA, dihydroxyacetone. So this is a keto triose. Keto triose 1, 2, and 3. And it is a keto triose. So this is the functional group and CH2. So this is 1, this is carbon 2, and this is carbon 3. Simple, and let's move to the next. So most common structure are the hexoses. The most common encountered sugar is glucose. Glucose, it is an aldohexose. Aldohexose, so aldohexose, let's draw the structure. 1, 2, 3, 4, 5, and carbon number 6, CH2OH. And it is an aldotriose, it means it has an aldehyde. Now we can draw this H, H. Now in case of glucose, this third OH will be on this side, and H, H, this is H. Now we can see on the third carbon, let's use a different color. So carbon 1, which will be an aldehyde group, 2, 3. So on third carbon, this will be on the OH, or third OH will be on the left side. 4, 5, and 6. So this is the structure of D-glucose. Now its respective keto sugar is fructose. fructose, it is again it is a ketohexose. So you know ketohexose. Hexose means you'll have to draw six. It is a six carbon compound. So let's draw this six carbon. 1, 2, 3, 4, 5, and 6. CH2OH. Now because it is a ketone group, so C2 is a carbon. C2 is the carbonyl carbon. Now C1 it will be CH2OH. COH this will be OH like the glucose. H H and balanced by the. Okay, simple. Now in this we can compare that glucose is a key. This is the structure of D ketose. Now the difference you just have to learn that okay the glucose first learn the structure of glucose. It is an aldohexose. Draw the six carbon, and the first carbon will be the aldehyde, and the three on third carbon the OH will be on the left side. And similarly, fructose is the ketohexose. The keto it will have a ketone group. The C2 will be the carbonyl group, and rest of the structure will be like similar to glucose. If you learn the structure of glucose, you can easily make out the fructose structure. Now let's learn the structure of, let's use a different color again, galactose. Now galactose the structure of from it is again an aldohexose. So its structure is quite similar to glucose. The difference you will see, 5. Now if you take a look at it, the structure is quite similar to glucose, except that at the C4 this OH is on the left hand side, and so the galactose differ from the glucose only at the C. So if you know the structure of glucose, you can easily make out the galactose structure. Now the structure of the mannose. Mannose is also an aldohexose. So again, you'll have to draw six carbon. 1, 2, 3, 4, 5. So it is like glucose only. See, it differs. It is like glucose. The configuration of HN of different carbon over here is like glucose only, except at the C2. Here this OH will be on the left side. So now you can compare these two structure, and you'll find that this mannose, the structure of mannose and the glucose are similar, except that if you know the structure of glucose, you can draw the structure of glucose, and at C2 this OH will be on the left side. This is where it differs. So here I would like to introduce one term, epimer. So if we compare the structure of this glucose and the mannose, you will find that they differ in configuration only at one carbon, that is the C2. So those those isomers which differ in configuration only at one carbon are called epimers. So glucose and mannose, glucose and mannose are C2 epimers. C2 epimers. Glucose and mannose, which means they differ in configuration only at C2. And see, glucose and galactose, they are C4 epimers. It means glucose and galactose. Glucose and galactose are C4 epimers. It means they differ in configuration only at C. Next is like you would have seen mannitol. You would have heard this term glucitol. Glucitol is also known as sorbitol. So this is an alditol, as we have talked in last video. It is alditol of glucose. So you need not to remember all these structures. So you just need to know that glucitol or sorbitol is actually an alditol of glucose. The structure will be similar to glucose, except that the aldehyde group is now reduced to alcohol group. So, and similarly, if we have this mannitol, it is the alditol of mannose. Mannose. So, if you know the structure of mannose, you can easily make out the structure. Now, you can just start, just learn first the structure of glucose. And in this, you know that glucose is an aldohexose. Hexose means you first draw 6 carbon. Aldohexose means the first carbon will be an aldehyde group, and then OH on the right side, because it is we are drawing the structure of D-glucose. Most of the carbohydrate, even all the carbohydrate found in nature are D form. So on third carbon, the OH is on the left side. Based on the glucose, you can draw the structure of fructose. On fructose is a ketohexose. All other configuration will be same. And if we talk about mannose, in case of mannose and glucose are C2 epimers, so they will differ at C2 carbon. Glucose and galactose, you just have to remember that glucose and galactose are C4 epimers, so they will differ in the configuration at C4. Okay, and glucitol is an alditol of glucose, mannitol is will be an alditol of mannose, so they will differ in this. Aldehyde will, instead of aldehyde group, you will write alcohol. So I hope it all will help in learning the structures of these monosaccharides. Okay, now we'll learn about how we'll make the cyclic structure. So if you know the linear structure of a molecule, you can easily make out the cyclic structure of a molecule. So before coming to the pyranose form, how we'll draw the pyranose form, just learn how this cyclic ring forms. In this linear chain, when there is an intramolecular hemiacetal formation, reaction takes place. This aldehyde group reacts with the OH group of the C5 or the C4. And if it reacts with C5, then a 6-member ring forms, and it will form the pyranose form. So this is how this cyclic ring forms, and the C1 attacks the OH of the C5. So if you know the linear structure of glucose, you can easily make out the structure of the cyclic structure. So let's say glucose mostly exists in the pyranose form. Let's make pyranose form. Pyranose form is a 6-member ring in which there are 5 carbon and 1 oxygen atom. This is the pyranose form in which this is C1, C2, C3, C4, C5, and when we have learned that this during the cyclic ring formation, the C1 reacts with the C5, so the C6 is out of the plane, out of the ring and out of the plane. This is 6 carbon, CH2. Okay, now C1, the configuration at C1, it can have an alpha configuration or beta configuration. So it could have like this. The OH could be on this side if above the plane. If it is on this side of the plane towards the 6 carbon, then this is the beta configuration. And this is a D glucose because this OH on the right side. So this is beta D. Now this is C1 and C2. So on C2 the configuration will be H and OH. On C3 the OH is on the left side, so on CO3 this OH will be here, H, H, and on C5 this will be H. So I think if you know the linear, if you can know the linear structure, you can draw the cyclic structure easily. So you can see over here also to the C, there is one C6 attached, CH2H group. It is here, C6, one H here, and in the hemiastrial ring, this will be attached to this. It is linked to the, and this O is linked to the C1. This is C1. This is the anomeric carbon. This is C1 is the anomeric. It can attain a configuration of alpha or beta by breaking of this bond. That's why this bond is often represented by a wavy line, because it can break and reforms, and the configuration of the C1 can be converted from alpha to beta. So I hope now you must be very familiar with most of the structure, and if you practice them, first learn, then you will be quite familiar with these structures. First learn the structure of glucose, the linear structure of glucose. From that draw the structure of fructose, galactose, mannose, and then try this. So how to, from the linear structure, how you can make the cyclic structure. Okay, now the ribose and deoxyribose, because these are also two very important sugars. Ribose is present in RNA, ribose is present in RNA, and it is present in DNA. If you can see, okay, what's the difference between ribose and okay, let's talk about the structure first, then we'll talk about the differences. Now ribose. First, let's learn about ribose. Ribose. It is an aldopentose. It's an aldopentose. Okay, aldopentose. So you can see 1, 2, 3, 4, and 5. There are 5 carbons, and it exists in pure. This is the pure. This is it exists in pyranose form. The C1 reacts with the C4. Okay, pyranose form means it will form a 5-atom ring. 5-atom ring, of which 4 will be carbon and one will be oxygen. So this is pyranose form exists like this. This is how you make the pyranose form. This is C1. Again, it can have an alpha or beta configuration. Okay, and this C2. The linear structure of ribose is like this. It is an aldopentose, first, second, third, fourth, and fifth. All OH on this side. So it is a D-ribose. This is a D-ribose. Now you can draw from this. You can draw these structures. The C1. This is the beta configuration because the OH and the CH2H are on the same side. And on the second, now we can draw this OH, and here you can draw the H. Also on the fifth carbon, so here the hemiacetal ring formation takes place by reaction between the C1 and the C4. So this C4 over here is linked to one this C5, this is this C5, and to this H, this is H, and O. It is linked to this O, and this O is linked to the C1. And this O is linked to C1. This is how you will make the pyranose form of the ribose. Now deoxyribose, there's only one difference how this deoxyribose differs from the ribose is on the second carbon there is an OH, and in case of deoxyribose it is H. So it is 2-deoxyribose. So this 2-deoxyribose means at the second carbon, D means removed or lacking, oxygen means oxygen. This oxygen at the second carbon has been removed. So this oxygen at the second carbon has been removed and only H is left. So it forms the deoxyribose. So because it lacks one oxygen, it is, first of all, it has obviously low molecular weight. It is more stable because it lacks oxygen. Presence of oxygen in this, it makes it more reactive. Okay, and lack of oxygen, it makes it more stable. So it is more stable, having low molecular weight. It is present in DNA and it is present in RNA. It has high molecular weight because of presence of oxygen, and it is less stable or more reactive. I hope now it clear this, or you will find the structure easier. So now you must be very familiar with the structure of the glucose. This is beta D glucose. So glucose, as you can see, this is the pyranose form. Okay, pyranose form means 6-atom ring of which one is oxygen and 5 carbon. This is C1, C2, C3, C4, and C5. This is C6. Beta form, why? Because this OH and this C6 carbon, they are on the same side. They're above the plane. They're on the same side of the plane. That's why it is a beta configuration. This is the structure of beta D glucose. Now beta D glucose 6-phosphate. So 6-phosphate means it has a phosphate group attached at the 6 carbon of beta D glucose. So its structure, you just have to draw the structure of glucose, glucose, and then beta form. So you'll make this OH on the upper side, and on the 6th carbon there is a phosphate group. So this is your 6th carbon, this is the 6th carbon, H2O. So to this you will attach a phosphate. Now attachment, why this addition of phosphate has biological importance. First of all, it activates the carbon. What is it is important in biological terms? It activates the sugar. Addition of phosphate to any molecule activates the sugar. In most of the biochemical, you will see that the sugar or the other molecule are first activated by addition of a phosphate group or UDP group or some ADP group. So they are first activated by adding an activating molecule. So addition of phosphate, it activates the molecule, and now it can participate in various reactions. Participate in various reactions, various metabolic reactions. For example, in glycolysis, you must have seen the glucose is first converted to glucose 6-phosphate, and in glycolysis, then it can enter either glucose 6-phosphate, then can enter either the glycolysis or glycogenesis. Okay, and another thing, this addition of this phosphate group imparts negative charge to it, imparts negative charge to the molecule, which prevent its exit from the cell, which prevents its exit from the cell. Because the cell membrane does not have a transporter for transporting this charged molecule. So it helps in retaining this molecule inside the cell. Okay, next is these are amine derivatives. So in many biological structure, we'll see this amine derivatives of the sugars. So this is glucosamine, beta D glucosamine. Beta D glucosamine. So you'll have the structure of glucose, and this is the beta form, and glucose. In case of glucosamine, you will see an amino group at the C2 carbon. So this is the how this glucosamine differs from the glucose. This is a derivative of glucose. Glucosamine, in there will be an amino group. Similarly, in galactosamine, there will be an amino group. This is the structure of galactose. If you remember, uh, we have learned that the difference between the galactose and glucose was at the configuration at the C4. So you can just check, you can just check for the configuration, and you will see the configuration of the C4. This is the C4. On all carbon, the configuration is similar except at this C4. So OH over here is on the above the plane, and here is on the is below the plane. So this is galactose. Okay, once you learn to draw the structure of glucose, you can actually make out the structure of galactose. Now galactosamine. So there is an amine group at the C2. Similarly, for the monosamine, there is an amino group at the C2. Okay, if you remember, the mannose and the glucose, they are C2 epimers. Okay, so they differ in configuration at C2. The OH over here in case of glucose, and in case of mannose, the OH will be on this side. So this OH is replaced by an amine. So this is the structure of mannose. So by this time you should make, once you learn the structure of glucose, all the structure are easy to learn. So now the amino sugar. So this is the structure of glucosamine, and where this OH at the C2 position is replaced with amino group. Now the amino group is often acylated. So this is the presence of acetic acid. So you see this CONH bond over here. So this is N-acetyl beta D glucosamine. So one should not be worried with the structure. How we will make it? just first of all, let's make the structure of glucose. Simply make the structure of glucose. The pyranose form like this. Okay, then we have glucose structure. This will be H. This will be. Now amino group in this OH is replaced with an amino group. So it will be NH2. So this has become glucosamine. Now N-acetyl glucosamine means N-acetyl. This N has been acetylated. So this will become, there will be an attachment of acetic acid. This will be NHCO. So it will now form N-acetyl. N-acetyl. It means there is an acetyl group attached to the N amino group N-acetyl beta. So this has a beta form. Beta, it is a D form, and glucosamine. Glucosamine. It means it is an amine derivative. So amine derivative means there is an NH group at the C2, and to this NH there is an acetyl. So this is N-acetyl beta D-glucosamine. It is, in short, it is called as glucnac. Glucnac. It is often present in the cell wall of bacteria, peptidoglycan. Okay, and glucnac, and there's another derivative, another sugar derivative present in the cell wall, that is called as murinac. So what is a murinac? So you see, this is a murinac where it is also an amino sugar. So there is an amino group at the C2, and this is muramic acid actually, and there at the C3 there is a presence of lactic acid. This is a lactic acid. So this is muramic acid, and murinac is when this amino group is acetylated. So this, when this NH group is acetylated, then it is called as N-acetyl muramic acid, and in short it is called as murinic. Murinic is also a component of the peptidoglycan. Okay, next is acetic sugar. So this is glucouronate form. In glucouronate is when this 6 carbon is oxidized to acid form. So this is the glucouronate form, and gluconate when the C1 is oxidized to the C1, that is the aldehyde group is oxidized to the carboxylic acid, then it forms the gluconate. Okay, when this acid, the C1, attacks the OH group, and then there is a removal of water molecule, and there is a formation of ester linkage between the C1 and the C5. The C5 is known as the delta carbon. So there is a ester linkage formation between the C1 and this delta carbon. So delta carbon. this delta carbon, there is an ester formation between the C1 and the delta carbon. So it forms D-glucono and delta lactone structure. Next, one more important acidic sugar is N-acetylneuramic acid. This is also known as a sialic acid. Most abundantly present in brain and an important part of there. So it is a 9-carbon sugar. It's a 9-carbon sugar, as you can make out. 1, 2, 3, 4, 5 in the backbone, and then these 3 and 1 this. So here there is a carboxyl group present at the C1, and at the C4 there is an amino group which has been acetylated, and then at the C5 there is an R group uh, which is this, which is a 3-carbon molecule. So this is the sialic acid structure, which is most abundantly present in the brain. I hope this video will help you in learning these structures. We'll talk about these structures of disaccharides and polysaccharides in the next video. So, and there are other things about carbohydrates. So please, if you want me to continue, please do subscribe and do share with people to whom it could be beneficial. Thank you.